In the early 1990s, Rachel Yehuda was running one of the first biological studies of PTSD at Mount Sinai Hospital in New York. She was studying Vietnam veterans, measuring their cortisol, trying to understand why their stress hormones looked wrong. Then the phone calls started. They came from the children of Holocaust survivors, not the survivors themselves, their children, adults who had never been inside a concentration camp, never seen the war, never experienced the trauma firsthand. And they were calling to say, "We are casualties too." They described growing up in homes saturated with grief, parents who couldn't sleep, parents who hoarded food, parents who flinched at the sound of boots on pavement. And these children, now adults in their 40s and 50s, were suffering from anxiety, depression, and PTSD at rates far higher than the general population. The clinical community had an explanation for this, a psychosocial one. Growing up in a traumatized household is itself traumatizing. You absorb the fear through the dinner table, through the silences, through the things your parents can't say. But Yehuda was a biologist, and when she drew blood from these adult children of Holocaust survivors, she found something that the psychosocial explanation couldn't account for. Their cortisol was wrong, not wrong in a random way, wrong in the same way as their parents. The same distinctive low cortisol signature that Yehuda had found in veterans with PTSD was showing up in people who had never been traumatized. Something had been transmitted, not through stories, not through parenting, through biology. And when Yehuda published this finding, the scientific community told her she was wrong. They said it was impossible, that trauma is psychological, that biology doesn't work that way, that you can't inherit an experience. She spent the next 25 years proving them wrong, and what she found didn't just change psychiatry, it changed our understanding of what inheritance means. Because your DNA is not your destiny, it's a library, and your ancestors' experiences decided which books are open. To understand what Yehuda found, you need to understand the most important revolution in biology since the discovery of DNA itself: epigenetics. For most of the 20th century, genetics told a simple story. You inherit genes from your parents. Those genes are fixed at conception. They code for proteins. The proteins build your body. End of story. Your DNA was your blueprint, and nothing that happened to you could change it. A parent who lost an arm wouldn't produce a child with one arm. What you experienced in your lifetime died with you. The genes went on unchanged. This was the central dogma, and it was wrong, not about the DNA sequence itself. That stays the same. You can't rewrite the letters of the genetic code through experience, but you can change which letters get read. This is epigenetics, the system above the genome, the layer of chemical modifications, methyl groups, acetyl groups, histone modifications that sit on top of your DNA and control which genes are switched on and which are silenced. Think of it this way: if your DNA is the complete works of Shakespeare, epigenetics is the highlighter. It doesn't change the words, it changes which passages your cells actually perform. Yehuda uses a different metaphor. She says genetics is the hardware, epigenetics is the software. Same computer, completely different behavior depending on which programs are running. And here's the part that shattered the central dogma. Your experiences change the software: stress, trauma, starvation, toxins, love, neglect. These experiences attach or remove chemical marks on your genes. They change what gets expressed, and some of those changes survive reproduction. When a cell divides, it doesn't just copy the DNA, it copies the epigenetic marks too. When sperm and egg combine, some marks get erased and reset, but not all of them. Some survive, some pass through. Yehuda has a metaphor for this too. She says, imagine a father giving his 7-year-old a sharp switchblade. In a peaceful suburb, that's a terrible gift. The child will probably hurt himself. But if that family is in a war zone, displaced, hunted, a switchblade might mean the difference between life and death. The biological changes she found in the children of Holocaust survivors are the switchblade. Not a curse, not a disease, a preparation. The parents' biology was trying to equip the next generation for a world it expected to be dangerous. The problem is when the world isn't dangerous anymore. When the war is over, but your body is still armed for it. Before Yehuda's work could be fully accepted, The scientific community needed proof that the environment could change epigenetic marks in humans, and that those changes could persist across generations. That proof came from one of the most horrific episodes of World War II, the Dutch Hunger Winter, November 1944 to May 1945. After the failure of Operation Market Garden, the Allied assault dramatized in the film A Bridge Too Far, the German army imposed a food embargo on the Western Netherlands. At the same time, the Dutch government called a national railway strike to support the Allies. The result was catastrophe. 4.5 million people were cut off from food supplies during one of the coldest winters in European memory. Official rations dropped to 400 to 800 calories a day. People ate tulip bulbs. They ate grass. They ate their own furniture for the calories in the glue. 20,000 people starved to death. And then, in May 1945, liberation came and the food returned almost overnight. This created something that would never happen in a laboratory: a perfectly defined experimental population, a genetically similar group of people, a precise start and end date for the trauma, excellent medical records maintained throughout by Dutch midwives and doctors, and decades of follow-up data. Scientists tracked the babies who were in the womb during the hunger winter, and what they found upended everything. Babies who were in the first trimester during the famine, conceived just before the worst of it, appeared normal at birth, normal weight, normal development. But decades later, as they reached middle age, they developed dramatically higher rates of obesity, heart disease, diabetes, and schizophrenia. Their bodies had been programmed in the womb for a world of scarcity. When that world never materialized, when post-war prosperity provided abundant food, the mismatch between their programming and their reality made them sick. In 2008, researchers at Leiden University compared the DNA of hunger winter babies, now in their 60s, with their same-sex siblings who were not exposed to famine. They found persistent epigenetic differences at a gene called IGF-2, a key regulator of growth and metabolism. 6 decades after the famine ended, the marks were still there. This was the first demonstration in humans that early environmental conditions could produce epigenetic changes that persist for an entire lifetime. But it got more disturbing. When researchers studied the children of the hunger winter cohort, the grandchildren of the women who starved, they found effects there too. Grandchildren of women who were pregnant during the famine were smaller at birth and had their own elevated health risks the famine had reached across generations, not through stories, not through parenting styles, through the molecular machinery of inheritance itself. A 2024 study confirmed that individuals exposed to the famine in utero showed accelerated biological aging 6 decades later, measured not by years, but by epigenetic clocks. Their DNA was aging faster than their bodies. The hunger winter didn't end in 1945. For the people who survived it, and their children, and their grandchildren, it never ended. The Dutch hunger winter proved that environmental trauma could change human epigenetics across generations, but it couldn't prove the mechanism. Humans are messy, too many variables, too many confounds. For that, science needed a controlled experiment. And in 2013, two researchers at Emory University designed one of the most extraordinary experiments in the history of biology. Brian Dias and Carey took male mice and exposed them to a chemical called acetophenone, which smells like cherry blossoms. Every time the mice smelled cherry blossoms, they received a mild electric shock to their feet. Within days, the mice were terrified of cherry blossoms. Standard fear conditioning. Nothing unusual. Then the researchers let the mice breed, and they tested the offspring. The pups had never smelled cherry blossoms. They had never been shocked. They had never met their fathers. In some cases, they were conceived through in vitro fertilization, eliminating any possibility of social transmission, and they were afraid of cherry blossoms. Not just a little. They startled dramatically at the scent. They were able to detect it at far lower concentrations than control mice, and when the researchers examined their brains, they found that the pups had more neurons dedicated to detecting that specific smell, more olfactory receptors, larger brain structures devoted to that exact scent. Their brains had been physically restructured by their father's experience, an experience that happened before they were conceived. And it didn't stop there. The grandchildren were afraid too. The epigenetic mark, a change in DNA methylation at the gene for the specific olfactory receptor that detects acetophenone, passed from father to son to grandson, a learned fear transmitted through sperm, inherited by descendants who had never encountered the thing they feared. David Sweat, a neurobiologist at the University of Alabama, called it the most rigorous and convincing set of studies published to date demonstrating acquired transgenerational epigenetic effects. The implications were staggering. If a mouse can inherit its grandfather's fear of cherry blossoms, what are humans inheriting from grandparents who survived slavery, genocide, famine, war, colonization? Carey Ressler became interested in this question for a specific reason. Before the mouse experiment, he had been working with impoverished communities in inner city Atlanta, where cycles of trauma, addiction, and mental illness seemed to pass from generation to generation with a ferocity that psychology alone couldn't explain. He suspected the cycles weren't just social, they were biological, written into the epigenome, inherited through the germline. The cherry blossom experiment didn't prove that in humans, but it proved it was possible. While Yehuda was studying Holocaust survivors and Dias was shocking mice, a Swedish scientist named Lars Olof Bigren was doing something extraordinary with parish records from a remote town in northern Sweden called Overkalix. Overkalix is above the Arctic Circle. In the 19th century, it was completely isolated. The population was genetically homogeneous. And because Sweden is Sweden, the record keeping was immaculate. Births, deaths, marriages, harvests, food prices, all documented going back centuries. Bygren realized he could use these records to ask a question nobody had ever been able to answer. Does what your grandparents ate affect how long you live? He tracked three cohorts of people born in 1890, 1905, and 1920, along with their parents and grandparents, and he cross-referenced their lifespans and causes of death against the harvest records from their grandparents' childhood. What he found was one of the most startling results in epidemiology. If a paternal grandfather had gorged during a year of exceptional harvest, a feast year right before puberty, his grandsons had a fourfold increase in diabetes-related deaths. Conversely, if the grandfather had nearly starved during that same window, his grandsons lived longer, significantly longer. When socioeconomic factors were controlled, the difference in lifespan was up to 32 years. 32 years of life, determined not by your own diet, not by your parents' diet, but by whether your grandfather feasted or starved as a 9-year-old boy in a parish above the Arctic Circle. The effects were sex-specific and line-specific. Paternal grandfather's nutrition affected grandsons, but not granddaughters. Maternal grandmother's nutrition affected granddaughters. The marks were traveling down specific lineages, following the germline, respecting the biology of X and Y chromosomes. This wasn't psychology. This wasn't parenting. This wasn't culture. This was biology, recorded in the molecular margin notes of DNA, passing silently from grandparent to grandchild across a century. A larger replication study using over 11,500 individuals from the Uppsala multi-generation study confirmed the core finding. Paternal grandfather's food access before puberty predicted grandson's mortality. The signal was real. Bygren published his initial data in 2001. When he presented it, many scientists didn't believe him. The idea that a single season of overeating in a child could kill his grandchildren sounded like a just so story, like folk wisdom dressed up in statistics. But the data held. And now it's been cited over 2,000 times. Because what Bygren found in Overkalix is the same thing Yehuda found in Holocaust survivors and Dias found in mice. The past is not past, it's in your cells. So how does it actually work? How does a grandmother's starvation rewrite her grandchild's biology? The answer centers on a gene called FKBP5. This gene encodes a protein that acts as a gatekeeper for the stress response. It modulates how your glucocorticoid receptors respond to cortisol. In plain language, FKBP5 helps determine how reactive your stress system is. In 2016, Yehuda published the landmark study. She examined FKBP5 methylation in Holocaust survivors and their adult children. Both groups showed altered methylation on the same region of the gene. But here's the twist that made the study famous. The changes were directionally opposite. Holocaust survivors had higher methylation at the FKBP5 site, Their children had lower methylation at the same site. The gene was being adjusted, but not by copying the parent's setting, by compensating for it. This is consistent with what happens during fetal development. If a mother with PTSD has low cortisol levels, the placental enzyme that normally shields the fetus from maternal cortisol may be disrupted. More cortisol reaches the fetus. The fetus's own stress system compensates by recalibrating its glucocorticoid receptors. The child isn't born with a copy of the mother's trauma. The child is born pre-adapted for a traumatic world. Stress system on hair trigger. Cortisol response recalibrated for danger. Yehuda found that maternal PTSD and paternal PTSD produce different effects in offspring. Maternal PTSD was associated with lower cortisol and greater glucocorticoid receptor sensitivity in children. Paternal PTSD produced different methylation patterns. When both parents had PTSD, the effects interacted in complex ways. This means the transmission isn't simple. It's not a photocopy of trauma. It's a biological conversation between generations, a negotiation between the parent's experience and the child's developmental environment. There are also behavioral pathways. In Michael Meaney's famous rat studies, mothers who licked and groomed their pups more produced offspring with different epigenetic marks on their glucocorticoid receptor genes, marks that made those pups calmer, less reactive, more resilient. Neglected pups got the opposite marks, and those marks passed to the next generation, not through DNA, but through behavior shaping biology, shaping behavior. Yehuda found that Holocaust offspring with mothers who had PTSD showed cortisol patterns consistent with maternal overprotection rather than neglect. The traumatized mother doesn't just pass trauma through the placenta, she passes it through how she holds the child, how she responds to the child's cries, how much fear she wraps around the child's world. Biology and behavior, epigenetics and attachment, two channels of transmission operating simultaneously, reinforcing each other across generations. This is why the old nature versus nurture debate was always the wrong question. The answer is neither and both. Nature and nurture are the same system, viewed from different angles. So, what does all of this mean? For you, personally, right now, it means your body is not entirely yours. It's a palimpsest, a document written over and over again by the people who came before you. Your grandmother's famine, your grandfather's war, your great grandparents' displacement. Some portion of their experience is literally in your cells, in the chemical marks on your DNA, influencing which genes are loud and which are quiet. This is not destiny. Yehuda is emphatic about this. Epigenetic marks are not permanent, they are responsive. The same system that allows trauma to be written into the genome allows healing to be written too. Yehuda's own research has shown that therapy, specifically EMDR and prolonged exposure therapy for PTSD, produces measurable epigenetic changes. The marks shift, the gene expression changes, the biology responds to the psychological intervention. You are not trapped by what your ancestors survived, but you are shaped by it, and understanding that you are shaped by it is the first step toward changing what it means. Think about what this implies for entire populations: descendants of enslaved people, descendants of colonized populations, descendants of refugees, descendants of famine survivors from Ireland to China to India to Ethiopia. Maria Yellowhorse Braveheart, a Lakota scholar, has spent decades documenting what she calls historical trauma in indigenous populations. The cumulative collective trauma of genocide, forced relocation, boarding schools, and cultural destruction. Her work shows that the psychological footprint of these events persists across generations in measurable ways. Now, epigenetics offers a biological mechanism for what she observed clinically. The trauma didn't just scar the culture, it scarred the cells. And this is where the science becomes political in ways that make some people deeply uncomfortable. Because if generational trauma is biological, if it's in the epigenome, then the consequences of historical atrocities are not just historical. They are present, they are measurable, they are in the blood of the living. But here's what Yehuda insists we remember. The epigenome is not a prison, it's a conversation. She says, we're starting to understand that just because you're born with certain genes, You're not in a biologic prison as a result of those genes. The marks can be changed through therapy, through nutrition, through stress reduction, through the very act of understanding what was done to you and choosing to respond differently. Healing is epigenetic too, and this brings us to perhaps the most profound implication. If trauma can be inherited, so can resilience. The same studies that found low cortisol in the children of Holocaust survivors also found something else: remarkable adaptive capacity, an enhanced ability to detect threat, a stress system calibrated for survival. The switchblade, remember? A terrible gift in peacetime, a life-saving one in war. The children and grandchildren of people who survived the unsurvivable are not broken. They are loaded, carrying biological equipment forged by their ancestors' worst moments. The question is not whether you have the marks. The question is what you do with them. In our last episode, Carl Friston showed us that every living system maintains itself by minimizing surprise, by building models of the world and acting to confirm them. Epigenetic inheritance is how those models get passed down. Your ancestors' model of the world, dangerous, scarce, threatening, is preloaded into your biology. Your job is to update the model. The contemplative traditions have always known this, that the past lives in the body, that ancestral patterns repeat until they are consciously interrupted, that liberation is not just psychological, but physical, a releasing of tensions held not just in your muscles, but in your cells. Yoga, breath work, meditation, somatic therapy. These practices aren't metaphorical healing. If epigenetics is right, they are molecular healing, rewriting the marks, updating the code, sending a different message to the next generation. You are not just living your life, you are editing a document that your children will inherit. If this journey into the biology of generational trauma changed how you think about yourself, subscribe to Consciousness Cartography. We're mapping the hidden forces that shaped how humanity thinks about mind, body, inheritance, and the nature of reality itself. The map continues.